Hi, everybody. My name is Paula Porto. I'm a Master of Soil Science student at the University of British Columbia. My supervisor is Dr. Sean Smuckler, and today I'm going to talk about the effect of soil drainage on soil greenhouse gas fluxes from highbush blueberry fields in Delta, BC. So how does water impact soil greenhouse gas emissions? We know that microbes and roots produce greenhouse gases when they respire in the soil, and that the magnitude and timing of these emissions can be impacted by a variety of factors, one of the main ones being soil water content. Um, Water impacts nutrient transport, and it also defines the type of biological respiration that takes place. When a soil is saturated, its pores are filled with water instead of air, so anaerobic respiration dominates, which produces nitrous oxide and methane. In unsaturated soils, there's more oxygen in the pores, so aerobic respiration takes place, and the product of which is carbon dioxide. In well-aerated soils, methane consumption can also occur. So these are just general trends, as we can see from this plot of nitrous oxide emissions over soil water content. Although generally total nitrous oxide production increases with soil water content, there are different pathways that lead to nitrous oxide production, and their rates peak at different soil water content levels. So reality is much more complex than these trends. My study region was Delta in the province of British Columbia. This is a productive agricultural region that has glaciolic soils, high water tables, and receives above 1,000 millimeters of rain per year, usually from October to March. In this region, the majority of farmers use some sort of artificial strategy, drainage strategy, to deal with the excess water in their fields, and 59% of farmers use subsurface plastic tiles. Given this context, we wanted to know whether drainage system and other soil and environmental variables impact soil greenhouse gas emissions. More specifically, we wanted to know if we could recommend a beneficial drainage management practice that could mitigate greenhouse gases. So to answer these questions, I conducted an observational study, including a total of 9 blueberry farms, each in one of 3 drainage groups. In the undrained group, farms had no artificial drainage strategy. They mostly relied on natural soil drainage. Fields in the drained group had plastic subsurface tiles installed, and then fields in the pumped group had plastic tiles in addition to ditch pumps. And if you're not familiar with plastic tiles, they're basically these perforated plastic tubes that are installed below ground at an angle, and they redirect water from the fields into an open ditch. That if the ditch system's working properly, then the ditch takes water away from the field as well. And the difference between the drains and the pump groups is that so when there's heavy rainfall, water sometimes rises above the level of the tile outlet, preventing the drainage tiles from removing water from the fields. So when this happens, farmers in the pump group can actually pump water out of the open ditch, allowing their tiles to continue to drain water. So I measured soil greenhouse gas emissions, CO2, methane, and nitrous oxide every 2 weeks for 2 years from 2017 to 2019 from each of these fields. I used a portable FTIR analyzer and non-steady state manual chambers, and I collected ancillary environmental data like soil water content and depth to water table. Before I went on with my greenhouse gas analysis, I plotted 2 soil drainage indicators by group. So in the top row here, I have water table depth over time. And on the bottom, I have volumetric water content taken at the surface over time as well. And as you can see, there's a lot of overlap among these groups. So it wasn't surprising that when I analyzed the data with the linear mixed effects model, that I found no significant effect of drainage group on soil greenhouse gas emissions after on cumulative emissions after 2 years. So this was not what I was expecting when I first started the study. So I went back to R and I used a principal component analysis to group the fields into two distinct hydrology categories. And I did this using two variables, depth to water table and water content at the surface measured as volumetric water content. And thankfully I was able to create two distinct groups. One I called the high water table group, and then the low water table group, each having 3 fields. And so I plotted the same variables, water table depth and volumetric water content over time, but this time by my high and low water table groups. And you can see that they're much more distinct. The high water table group, as it is called, has a higher water table generally throughout the sampling period, and also a higher volumetric water content. So fields in this group are generally wetter. And when we look at daily greenhouse gas fluxes, I know this is a bit of a busy slide, so bear with me. In the top row here, I have CO2 emissions as kilograms of CO2 per hectare per day over time. The shaded bands here represent the growing season from April to the end of September. My second plot is showing nitrous oxide, the third one showing methane, and the final one is showing average weekly temperature over time as a line, and then total weekly precipitation as the gray bars. So when we focus here on carbon dioxide, we can see that it sort of follows the same pattern as the average weekly temperature line. In the growing season, the emissions are higher, and then they're lowest in the winter months. And you can see that the high water table group, which is in dark blue, is generally, it either matches emissions from the low water table group or it's higher than that group. And then for nitrous oxide, we can see that there are peaks, spikes happening in the fall and the winter for both years. And for methane, there's a really large spike here happening in the winter as well. And so when I zoom in and I change the scale of these two graphs, I'm just zooming in here, we can see that there are also other nitrous oxide spikes happening from April to June, which is when the farmers usually apply their split nitrogen fertilizer applications. And then for methane, we see that there's some consumption happening and some other spikes, but generally emissions average around 0. And here in this slide, I'm showing cumulative emissions by season. So in the top row, I have year 1. In the bottom row, I have year 2. On the left, I have carbon dioxide, in the center is nitrous oxide, and on the right, I have methane. And the color coding is the same. So dark blue for high water table and light blue for low water table. So for carbon dioxide, we see that it follows the same pattern across the seasons and also for both years, with the high water table group emitting more carbon dioxide than the low water table group. And we actually get a significant group effect for both springs. And then for nitrous oxide, we also see a similar trend with the high water table group emitting more nitrous oxide. And in the fall and winter of year 1 and in the fall of year 2, we get a p-value below 0.1, and then in the winter we get a p-value below 0.05 in the winter. For methane, we didn't see any significant group effect on emissions. We see that in the winter of year 2, we had our highest cumulative seasonal emission. And when I zoom in here, I'm also changing the scale again here, we see that the emissions don't really have a consistent pattern across the seasons and across each year. So there's more variability with this gas. In this slide, I'm showing cumulative emissions by year, with year 1 on top and year 2 at the bottom again. And so it's following the same trends for carbon dioxide and nitrous oxide, with the high water table group emitting more. And we get a significant p-value for carbon dioxide in year 2 and nitrous oxide for both years. And then for methane, again, no significant group effect. And we see that in year 1, on average, the fields were consuming methane, but in year 2, they were emitting methane. In conclusion, my study indicates that poorly drained fields emit more carbon dioxide and nitrous oxide than better drained fields. On average, each year, the high water table group emitted 27% more carbon dioxide and 146% more nitrous oxide than the low water table group, which indicates that effective artificial drainage has the potential to reduce soil greenhouse gas emissions. However, more research is needed to elucidate the impacts of soil drainage on emissions. For example, we'd like to know if the patterns are consistent across different soils and climatic regions, and it would be interesting to see a full greenhouse gas budget for this agroecosystem. For next steps, I will analyze the impact of other variables in soil emissions, such as thickness of sawdust mulch, and use the process-based denitrification decomposition model to simulate soil emissions from highbush blueberry farms in our region. I'd like to thank our funder, Agriculture and Agri-Food Canada, and everyone that's helped me get here. Thank you so much.